My name is Sean Craig. I'm a professor at Cal Poly Humboldt. It is my pleasure and honor to introduce the next set of speakers. We'll get right to it here. The first speaker is also from Cal Poly Humboldt. Marina Storey will tell you about her research entitled Hold the Salt. Are there refuges from predation by purple urchins on kelp near the river mouths?
I feel like I pressed a button accidentally. Can you guys hear me okay? Okay, then let's get started. So, as Sean said, hello everyone. My name is Marina Storey, and I'm a master's candidate at Cal Poly Humboldt, studying persisting kelp beds at the mouths of rivers in Mendocino County, with the hypothesis that the decreased salinity in these areas is negatively impacting purple sea urchin grazing on the kelp. Do I say next slide to you? Okay, thank you. So, little outline for what I'm going to talk about, background on kelp and urchins relative to salinity, and my hypothesis and how I'll answer that with my methods in the field and in the lab, the results I have thus far, and the conclusions I draw from that. Next slide, please. So, a bit of background. Kelp is this really important species that provides a lot of habitat and food for other very important species that culturally, economically, and in the kelp forest ecosystem. But unfortunately, around the world, we've seen loss of kelp in a lot of places, including in Northern California, where we've seen over 90% loss. Next slide, please. And so specifically for Northern California, these are our particular anthropogenic stressors that threaten our kelp. And so starting in the mid-2010s, from around 2014 to 2016, we had warm water anomalies that raised our sea surface temperature that desiccated our kelp. Around the same time, we experienced sea star wasting disease that ran rampant through all of the West Coast and decimated sea star populations, and especially the Pycnopodia halianthoides, also known as sunflower sea star, which is a known predator of purple sea urchins. And then around the same time, we saw purple sea urchins increase their population. And then with the lack of predatory sea stars, their feeding behavior changed from this passive behavior to an active feeding behavior. And without kelp, and with all these sea urchins around eating what kelp was coming back, there was no food for the abalone, and so they ended up starving to death, and the fishery closed in 2018. Next slide, please. So, about urchins, we just went over, they like to eat kelp. Their population has increased a lot. Their feeding behaviors have changed. So what can we do about this, right? Well, luckily, they're in a echinoderm. And so what that means is that they use a water vascular system, or they don't osmoregulate. So they're at the mercy of the conditions of the water around them, because they take the seawater surrounding them and they pump it through their system for physiological function. So they're particularly sensitive to changes in seawater chemistry, like changes in salinity. Next slide. So with all this information, people have been, and on top of this, people have made anecdotal observations of persisting kelp at the mouths of rivers in Mendocino County. So these are screenshots I took from Kelp Watch from 2020 to 2022. I believe it's the spring quarter. And so I added some arrows to point out the dark patches, which Kelp Watch uses satellite data to map out kelp. Next slide, please. The same creators of Kelp Watch came out with a paper recently that identified that the north coast of California is a climate refuge for kelp, which is where these rivers lie in my study. Next slide, please. So again, my question is to address if the decreased salinities at these river mouths in Mendocino are negatively impacting purple sea urchin grazing. And so I'll answer this in a combination of laboratory and field observations. So in the lab, I will test if constant salinity impacts urchin grazing, if intermittent pulses of decreased salinity impact urchin grazing, if decreases in salinity impact urchin mortality. And then in the field, I'll make observations about if sea urchin populations differ at river mouths versus non-rivering sites, and if the population size distribution, so how big or small these urchins are at river sites versus non-rivering mouths, and if so, how. Next slide, please. So my site pairs, starting with top left, I have the Noyo, so rivers are in blue and the reference sites are in red with the marker. So we have the Noyo River with its reference site Hare Creek, the Big River with its reference site Mendocino Headlands, also known as Pipeline, and then we have the Albion River with its reference site Dark Gulch, and the Navarro River with its reference site Saddle Point. And that's me and my old lab partner and dive buddy Duncan Jackson. Next slide, please. And so the way how we made observations is that we ran 8 transects either in a persisting kelp bed or around a CTD that we deployed in these reference sites and persisting kelp beds. And so it's basically a big measuring tape, and then we measure, we did 5 by 2 meters, so a meter on each side of the tape. And we counted bull kelp and purple urchins along with other species that could impact their abundances, for instance, like red urchins that may outcompete or something. And then we also sized urchins, or up to 30 individuals per species of both red and purples. And then the CTD loggers, that stands for conductivity, temperature, and depth. And so conductivity is the proxy for salinity in this case. And they take readings every 6 minutes, so we have a pretty refined data set for that. Next slide, please. And so this is what one of our sites looks like. And so this is at Albion River. The top is temperature and the bottom is salinity. And I added 2 bars here, one at 20 PSU and one at 15. So we can see that salinity drops below 20 PSU pretty regularly, and then also occasionally below 15 PSU. Mind you, ambient or regular seawater salinity is in the 30s, and so at our lab it's around 32 PSU in Trinidad. Next slide, please. So getting into my methods, for my lab experiments, I conducted these feeding experiments where I took this acrylic aquarium and put it in a water bath, so temperature was constant, with 1 urchin per tank with its kelp. And I examined salinities 32 to 10 PSU in increments of 2. My control is kelp without an urchin to measure change in kelp weight without grazing. And then I starved urchins 24 hours before the start of these experiments. All kelp and urchins were collected from Trinidad in California. And I conducted 2 style laboratory experiments, pulse and press. Next slide, please. So for my press experiment, this is that constant exposure to decreased salinities. So after the 24-hour starvation, I monitored their feeding rates for 72 hours, where I was taking a measurement every 24 hours. And then I did this by taking the kelp out, blotting it dry with a paper towel, and then putting it back in. And then my equation for adjustment for control is calculated by the urchin, or the kelp that was given to the urchin, multiplied by the control coefficient, which is the control's final weight divided by the initial, and then you subtract that by the final weight of the kelp that was given to the urchin. Lots of math. Next slide, please. And so for my pulse experiments, these are done a little differently. And so I did manual water changes every 6 hours, which I'm still doing today even. And so the way how I conducted this experiment was after the 24-hour starvation. For 24 hours, they were at 32 PSU, and I measured 4 points every 6 hours to get a baseline of their grazing rate. And then for 5 days, I pulsed them. So that's interchanging between ambient salinity at 32 PSU and that decreased salinity. And then after 5 days, I did another 24 hours at 32 PSU, measuring once every 6 hours. And then after that, I did 7 days of just measuring once a day to sort of see if they recover, basically, from that pulse effect. And then kelp was weighed. Kelp weight was the same, except a new piece of kelp was given every 6 hours with the water change. Next slide, please. And so we'll get into the results, starting with the field observations. I'm missing a title up there, but it's okay. So over here, we have algae on the left and invertebrates on the right. And this goes by site pairs. So at the top, we have Noyo River and Hare Creek. Second down is the Big River and Mendocino Headlands or Pipeline. The third down is the Albion River and Dark Gulch. And at the bottom, we have the Navarro River and Saddle Point. And so the main observations I have here was that Albion and Noyo were inverse of their reference sites, where they were dominant algae and lacking invertebrates, and their reference sites were the opposite. And then Mendocino Headlands, Big River, Saddle Point, and Navarro had an assemblage of both invertebrates and algae, but the overall themes that we saw was that the rivering sites had more algae and the reference sites had more invertebrates. And so I think I forgot to mention, but the blue columns are the rivers and then the red columns are the reference sites. And next slide, please. I don't know if you guys noticed, but there's no purple urchins on that last slide because the scale is too large and you wouldn't be able to see anything else. And so here, what I have is graphed again, adult bull kelp per site, and then purple sea urchin density per site. And so you can see here that there is some bull kelp at these reference sites and at all of our rivering sites. But then when you look at purple sea urchins, it's in the hundreds of individuals per meter squared, and it's a lot more at the reference sites and almost none, and some have none at the rivering sites. And I found that there is a statistical difference between river sites and reference sites for purple urchin densities, but no statistical difference for bull kelp density. Next slide, please. And so, looking at the size distribution of these urchins, we were lacking in urchins for the river sites to begin with. But on top of that, we're not really seeing 1 to 2 centimeter urchin size classes. And so typically that is what we think of as the new recruits as the year. That sort of smallest size class, they're between 6 months and a year old. Or it means that they have high post-settlement mortality rate, meaning that maybe the urchins could get there, but they're not living long enough. So next slide, please. Whereas with the reference sites, we see lots of urchins for both reds and purples. And in particular, we have 2 sites here at Mendocino Headlands and at Hare Creek that have lots of 1 to 2 centimeter urchins, indicating that there is recruitment occurring at these sites. Next slide, please. And so getting into my laboratory experiments, this is the press I did. So figure A, that is graphed with the salinity and how much biomass in grams the urchin consumed for 24 hours. B is at 48 hours, and C is at 72 hours. So you can kind of see that as you increase in salinity, you increase with the you increase in the grams consumed by the urchins. And all these graphs are statistically significant. Next slide, please. But that might be because all of the lower salinity urchins died after 24 hours. And so I included a black bar here at 18 PSU because all of the urchins from 10 PSU to 18 PSU died after 24 hours with a constant exposure to salinity. And then I had this red bar here at 20 PSU at the 48 and the 72 hours because after 48 hours, 2 of my 20 PSU urchins died and another one died at 72 hours. I'll also include some observations I made where 2 of my 22 PSU urchins and 1 of the 24 PSU urchins was trying to escape their tanks during this press experiment. Next slide, please. And then this is the results of my pulse. I graphed it differently because I felt like it was a lot easier to see. And so due to capacity reasons, I only did 6 of these salinities versus the full 12. And so up top, we have the lower salinities, which is 12, 16, and 20. And at the bottom, we have 24, 28, and 32 PSU. And so similarly to the press experiment, all of my 12 and 16 PSU urchins died in this experiment, and I've had 1 20 PSU urchin die. And overall, you can see with these bottom graphs here, they not only eat more, but at the tail end of these graphs, they uptick higher. I know that's not fair to 12 and 16 because they're not alive, but you know. And we'll have more stats coming soon if you come to WSN. Next slide, please. And then I wanted to include some photos of other behaviors that I collected. And so on the far left here, we have detached tube feet. And so he's got his tube feet out, but he's just floating his tube feet. Another style I saw was where their tube feet were completely retracted, and that's more similar to the second one here, but he's also spawning. And so that's a stress response that is common in invertebrates. And then this one second to the right here is got an enlarged Aristotle's lantern area. So that's their mouthpiece on the urchin, and it's got a lot of flesh there. And then this urchin here to the far right is dead. Next slide, please. So my takeaways from this to answer my questions that I proposed. Yes, purple sea urchin populations are significantly different at river mouths compared to reference sites. There's basically no urchins at these river mouths and a heck of a lot at these reference sites. Purple sea urchin size populations do differ, not only because there really isn't any urchins, but because there is no 1 to 2 centimeter urchins that would indicate that there is recruitment occurring at the river mouths, or it means there's high rates of post-settlement mortality. And then for my lab experiments, for both press and pulse, I would say yes, lower salinities do impact purple sea urchin grazing rates, because as you go lower in salinity, you have less consumption of kelp. And then I would say yes, lower salinities do directly increase purple sea urchin mortality, as seen in my results for both press and pulse. Next slide, please. And so just give big thank yous to everyone for funders and collaborators and my undergrad assistants, and for California Seaweed Festival for having me come here and talk. And that's my email up there. And so similarly to my good friend Brian, I am also looking for a full-time job. Thank you. Oh, should I wait for questions?
I don't think we've got enough time, but thank you. Thank you. We'll have to wait on questions, and I'm sure Marina will be glad to answer those very soon. So our next speaker is Julieta Gomez. She'll talk about kelp restoration, bull kelp, at the Greater Farallones National Marine Sanctuary.
Hello, sorry for the technical difficulties. Hello, my name is Julieta Gomez, and I am a kelp restoration specialist with the Greater Farallones Association. So it's a nonprofit based in San Francisco, and I'm here to talk to y'all about our bull kelp restoration efforts happening within the Greater Farallones National Marine Sanctuary. And this is a collaborative project with the Greater Farallones and Cordell Bank National Marine Sanctuaries, as well as Sonoma State and Moss Landing. And we use lab space at Bodega Marine Lab at UC Davis. Next slide, please. So I wanted to go over the importance of kelp forests, which I'm sure all of you have heard this all day, so sorry to talk your ear off about it again, but it enhances biodiversity and supports the multi-million-dollar fish industry, which relies heavily on healthy kelp forests. It also protects shorelines by dampening effects of large storms, and promotes ecotourism by providing luscious areas for scuba diving, kayaking, as well as whale watching. Next slide, please. So bull kelp is the species that we focus on to restore in this area. It is found from Central California all the way to the Aleutian Islands in Alaska. And its morphology, as we can see in this image, it has a holdfast, so that's like its root-like system, and that's how it attaches to the seafloor. And then it has a long stipe, which is similar to a tree trunk. And at the top of that, we have a nematocyst, or a gas-filled bulb. So then from that bulb are the blades, and that houses the gas exchange, photosynthesis, as well as the reproductive material called the sori. So that, or the dark rectangular patches in that image. Next slide, please. So bull kelp exhibits an alternation of generations life history. So this means that it has a microscopic stage as well as a macroscopic stage, and it is an annual species. So all of their growing happens during one season. So with the macroscopic stage, it starts off as a little spore, which are just circular microscopic things, and then it forms into gametophytes. So it'll form into female and male gametophytes, and then it'll reproduce sexually and create the sporophytes, which look like little corns on the cob. And then those sporophytes will grow and become the macroscopic sporophyte that we are used to seeing out in the ocean. And then from there, the sori will grow on the blades. Next slide, please. So, like I mentioned, they are an annual species. So starting off in the spring, they'll be macroscopically visible out in the seafloor. And then by summer, they reach the surface. And in fall, they are getting reproductive and sloughing off those sori. So the sori are negatively buoyant, and they are sinking to the seafloor and releasing all those spores. And then by the wintertime, they're doing their little microscopic thing and reproducing, and then growing and being visible by the springtime. So one thing that we're trying to achieve is have these year-round cultures in labs, so that way we can manipulate that timing of their life history. And that way we don't have to rely on that natural stock. So instead of having to wait until summer and fall when the sori are reproductive and having to collect those then, and then waiting 2 to 3 months for them to be reproductive, and outplanting in fall when it's too late, we can manipulate that timing by having all that stock ready for us in lab and being able to outplant in spring when it's ideal for their growth cycle. Next slide, please. So I co-manage this project with Rachel Carm from Sonoma State University, and we have various life history stages that we have at the Bodega Marine Lab. One of those are adult bull kelp crowns. So these crowns were actually received from our partners at Moss Landing Marine Labs. They got the sori originally from Russian Gulch in Northern California, up in Jenner, or down in Jenner. And then they gave us the juvenile sporophytes back in January, and we have grown them all the way to adulthood, and they are now reproductive. So we are using these sori to inoculate tanks for the next generation of lab-grown bull kelp, as well as for twine that we will outplant, and I'll get into some of our restoration efforts. Next slide, please. So inoculating tanks is way easier when you have the sori in-house and you don't have to go out to the ocean and boogie board to go get it. So what we do is we go to our tanks and we'll try to get the darkest patches of sori available, and we'll do a little iodine rinse to get any animals that may be growing off, and then we'll do a little seawater rinse to get the iodine off. And then we'll let the sori dry out for about 2 hours, and then after that period is up, we'll put them in a seawater bucket. And going from dry to wet will basically stress them out into releasing their spores. And then eventually we have this dark solution, spore solution, which is kind of like agua tamarindo type of color, and then we'll use that to put into our tanks. So then we'll inoculate tanks for the next generation as well as for outplanting. Next slide, please. So we have inoculated twine. That's one of our restoration methods. So we have all our tanks set to a 12-hour photo period as well as with filtered seawater. And we are using twine because there has been past restoration efforts where they've used pavers, which are stepping stones that are about 3 by 3 inches, as well as green gravel. But we found that those methods take up a lot of lab space and are kind of a safety hazard when diving, because you're just diving with like a bag of rocks, you know? And then we found that twine is easier to manage underwater and takes up less space in labs, so it's just more ideal all around. And specifically, we use this Japanese twine called Kuromona because there's been research actually happening here at Cal Poly Humboldt where they found that that is kind of the more ideal twine based on other materials. So we inoculated this twine in April 15th of this year. If you can go to the next slide. And this was on May 31st of this year when we saw our first visible sporophyte, and Rachel and I were super excited. It's just a few millimeters big. And then if you go to the next slide, then by July 8th, they got a lot bigger, and we were like, okay, we're ready to outplant. So we outplanted in July 24th, and later on in the presentation, when I talk about the outplanting efforts, you'll see how they're doing currently in the ocean. Next slide, please. So like I mentioned, we're also inoculating for the next generation of lab-grown kelp because we really want to close that life cycle and just have a stock that we have available to us year-round. So we inoculated on April 15th as well, and by May 20th, we were seeing kelp growing on the side of the tanks as well as on the airline, which is that line that you can see there. So they're just a few centimeters or few millimeters big, and then by June 10th, they were like centimeters big. So once they were that big, they were ready to graduate into their bigger tanks. Next slide, please. So we have these garrafones, or like 5-gallon water jugs, that are our carboys, or our tumble cultures. And we got this idea and help from our collaborators at Moss Landing Marine Labs, as well as Kalani from Scripps, Wow, she was just on cue. She just showed up. So we have our more adult juvenile, or our bigger juveniles in those tumble cultures, and then eventually they are moved outside to our outdoor tanks. Next slide, please. So this image shows how some of the juveniles look. So they grow their little nematocysts, and once the nematocyst is big enough, we'll move them outside and we'll crown them, which basically just means cutting the nematocyst in half so that way they can tumble freely in the tank and not just float at the surface like they would in the ocean. Next slide, please. So getting into outplanting, now that we've talked all about culturing, we work within the Greater Farallones National Marine Sanctuary, which you can see in that tiny map inlet. So the GFNMS encompasses 3,295 square miles of a nationally significant marine ecosystem. So that includes endangered species as well as threatened species. And we're specifically working within 2 sites, so Timber Cove and Fort Ross Cove. And as I'm sure you've heard in a lot of other projects this year, or a lot of other projects, there's a lot of permitting delays. There's also limitations with resources. So for us, we had a lot of vessel issues that we had to deal with. So that's why we're only focusing our efforts on 2 sites. Specifically, most of our efforts have been going in at Fort Ross Cove. So all these sites are within the GFNMS, and they're in, I guess not as north as we are here, but Northern California. Next slide, please. So we have a restoration area of 30 meters squared at both of these sites, and we have 9 modules or replicates where we are implementing 2 different outplanting methods. So we have 3-meter lead lines that are T-shaped, and they are clipped into eye bolts that are semi-permanently drilled to the reef. And the clips allow us to remove those lines at the end of the season as well as the beginning of the season. Next slide, or next thing, please. So we have a control line, and this allows us to see if there's any natural recruitment happening in our site. Next. And we also have the twine. So the outplanted twine is spooled around this semi-permanent line, and it has a little buoy to protect it from grazing. Next line, please. And we also have sorrel bags. So sorrel bags are these crab bait mesh bags where we put Storey, and that allows us to just have kind of like natural spores that we are releasing to just replenish that stock available. And for next year, can you click again? We will add spliced juveniles. So our partners at Moss Landing Marine Labs in Sonoma State have tried using spliced juveniles in their project in Mendocino, and they've found great success. It allows them to have the bull kelp grow up to the surface faster because they're already outplanting it at an older stage, and it allows you to like track how much growth you're having because you know exactly how much you outplanted, so you know how many should be there and how well they're surviving in the wild. Next slide, please. So this is some images of all the methods that I talked about. So the spliced juveniles are just, like I mentioned, just directly put into that line at an older stage. And the twine is the middle, so you can kind of see the buoy in the middle is the red thing, and that's to protect it from grazing. And this black bag is the sorrel bags. Next slide, please. So in order to outplant successfully, we also need to reduce that grazing pressure. So we work with 5 local commercial urchin divers, and they have been doing amazing work and have been trying to help us reach our goal of 2 urchins per meter squared. So at Fort Ross specifically, before we started outplanting, the urchin density was about 29 per meter squared, and currently as of September, it was down to 10 urchins per meter squared. So there's still a lot of work that needs to be done, and the urchin removals need to happen consistently in order to really get that encroachment down and really protect our outplants. Next slide, please. So this is what the outplanted twine looks like. So as you can see, there's fouling or growth of other species, like kind of on the side, like other browns. But in the center, you can see the little young bull kelp sporophyte. So it's super exciting. We definitely started a little late in the season, so the hope is to start earlier next year. But this is still a great increase from last year for our project specifically. Next slide, please. So for next steps is to grow our bull kelp sporophytes in the lab all the way to adulthood. We want to fully close that loop, and we're pretty close to doing that. We have a lot of juveniles right now that have pretty long blades but aren't reproductive yet, and we want to continue culturing that through the winter. And for next year, we're hoping to outplant early in the spring versus later in the summer, and add those spliced juveniles as a third restoration method. And hopefully we'll slowly watch our kelp forest come back over time as the project continues to grow. Next slide, please. So I want to say thank you to the funders, because all this project wouldn't be possible without all the money, of course, as well as our amazing teams and collaborators all listed here. So thanks.
Okay, thank you so much, Julieta. Moving on, we've got next talk by Kelly Morgan, and they will talk about an apprenticeship in Maine aquaculture.
Thank you so much.
You betcha. Let's make sure those slides are going.
Can everyone hear me okay? Sound good? All right. So my name's Kelly Morgan, and I'm going to be presenting on the Maine Aquaculture Apprenticeship Program. But before I do, I just want to say thank you so much to California Seaweed Festival for inviting me to speak. I don't know if we have any reps in the room, but it's an absolute dream to be up here with other scientists doing important work for our kelp forests. And it's a real pleasure. So my goal in this presentation is really just to share my journey into aquaculture and what aquaculture looks like in some other parts of the country to maybe inspire ideas in you all. Next slide, please. I grew up on the coast of Southern California and was fortunate enough to sail and surf and dive in our kelp forest there as a kid. And it's certainly this childhood that inspired me to go on and study our marine environment. And so I first have to give a shout out to my dad's here today. He's my dive buddy and my kelp buddy, and I definitely wouldn't be here without him. So thank you, Dad, before I move on. And so, like I said, this childhood definitely inspired me to study marine biology, and I went on to UC Berkeley, majored in integrated biology and marine science, go Bears. And this was in 2017, so just a few years after we saw some of the El Nino marine heat waves, endearingly coined the Big Blob, make landfall and destruct a lot of our kelp forests off the Pacific Northwest coast, right? It was also during this time that I, diving off the Sonoma coast down south, saw an urchin barren for the first time and saw that kind of devastation. Next slide, please. And my professional career took me sailing across the world, and one of these places I sailed pretty frequently was the Gulf of Maine. And so for those of you who don't know, the Gulf of Maine is one of the fastest warming bodies of water on the planet. And like everywhere else in the world, we're seeing record-breaking temperatures every summer, and coastal ecosystems are changing dramatically. Scientists and fishermen alike are seeing fisheries decline, and specifically lobster move further offshore. Lobstermen are having to steam further and longer to get the same catch that they needed to sustain their business and have been for generations. So it's with the help of a lot of advocacy organizations on the East Coast that are offering grants and loans that fishermen and lobstermen are turning to aquaculture as a more secure and stable job alternative to chasing after these wild fisheries. Next slide. Is it going to play? Maybe try clicking again? That's all right. It's actually just a lot of nice scenery of the East Coast. So if it plays, that's all right. But if not, maybe if you click on it, it might take you to the YouTube link. I'm not sure. Well, imagine a lot of beautiful videos of sea farming on the East Coast. And that was my segue to say there are over 200 sea farms out in Maine employing over 700 people year-round. And 1 in 6 of those farmers also hold a lobstering license. Most farms are cultivating oysters, but we also farm blue mussels, kelps, and other seaweeds, sea scallops, and there is an Atlantic salmon farm. There is some experimentation with clams, urchins, and American unami, also in Maine, which is pretty cool. And then there's several companies working to generate sea farming as a viable option for fishermen, and one of those is Atlantic Sea Farms with Bree Warner, providing sea farming as a job alternative during the winter, an alternate source of income for lobstermen. There's a cool company called Running Tide that was selling carbon credits to big corporations to fund large-scale sea farming projects. And then, of course, Green Waves down south in New York, educating fishermen on how to make the switch and become farmers. So when it came time for me to make a career pivot, I thought this is a field where organizations are putting science into practice and marketing the carbon drawdown potential of kelp, building these regenerative farms that have a positive impact on the environment they're in, and creating jobs for people who love working on the water and have for generations. And so I really wanted an in on this industry, and it was about this time last year that I stumbled on the Maine Aquaculture Apprenticeship program. Next slide, please. I also have my laptop here if we want to plug in, and I do have another video. It's not too much to ask. Let's see, is that gonna, whoop. And that's it, folks. So thank you so much. Here, 1 second. Hold this straight.
I think they got it, Kel.
It would be awesome for you folks to see the video if it is possible. No? All right. Well, that's okay. I'll tell you guys what to look up on YouTube. Okay, so the Maine Aquaculture Apprenticeship Program was started by Chris and Marin at the Gulf of Maine Research Institute and Christian Braden at Maine Aquaculture Association. And they saw a need for skilled laborers in a growing industry, and so designed this program to do exactly that. In order to graduate from the program, you need 2,000 hours of time on the farm and 144 hours of coursework. So this can take anywhere from like a year to a year and a half to complete, depending on the seasonality of your farm. But once you do, you graduate with a certificate in the trade of aquaculture that is nationally recognized and, at least right now, one of a kind. Next slide. So the coursework includes a 1-week intro boot camp into the industry, where we do everything from learning to tie a bowline to driving a small boat to what is the scope of aquaculture in Maine as compared to the rest of the country. We get tours of all the host farms. So we get to see all the different operations and how they differ from our own. We do a 1-week deep dive into the industry where we learn kind of more intensively about shellfish and seaweed farming in the country and in the world. In March, we're going to do a 3-day intensive outboard maintenance and troubleshooting workshop, which is pretty cool. And we're going to tour a few different hatcheries and processing facilities in this state. So it's all hands-on, very practical experience. Next slide, please. This is my cohort of apprentices. We're the second cohort to go through the program, and there's 9 of us between 6 different farms. So most of our time throughout the year is spent, 8-hour days, 5 days a week on the farm, and the classes serve as a sort of touch point for all of us to hang out, which is cool. And like I said, in addition to this trade certificate that we graduate with, we also gain experience in the industry and access to a wide network of stakeholders, from farmers to researchers to NGOs and the Department of Marine Resources. And so from the first cohort of apprentices that just graduated this last September, most have gone on to manage their farms. One apprentice is working for Maine Aquaculture Association itself. I know another one of them is interested in going to law school and studying marine policy. But what's cool about the program is even if we don't continue to be oyster farmers after our first year, it provides on-the-ground experience and an intimacy with the working waterfront that's attractive to a lot of companies in the sector. Next slide, please. So I work at Ferta Farms, which is comparatively the smallest farm of the group, and it was started by a father and son, Max and Chris Burtis, 6 years ago when Max was in high school. And they're very committed to pushing the aquaculture industry in a positive and mindful direction in conjunction with the Island Institute, which is a local advocacy organization in Maine. They've run analysis on the carbon footprint of our farm and actively work towards ways to reduce this impact. All of our gear, our tumbler, and everything on our workflow is solar powered, and we're hoping to get an electric outboard for our boat in this next year. This summer, we even experimented with floating buoys made out of mushrooms created by mycologist Sue Van Hook as an alternative to plastic buoys on oyster gear. We're supporting several research projects, including a master's student from Maine, sustainable aquaculture grant projects. And so this next video I was going to play, you can go to the next slide. It just talks about the impact of the apprenticeship program on furtive farms, and it's backed by some really emotional music, and you get to see Max like 3 years ago in his early stages of starting the farm, and it's very inspirational. It's all to say that they really could not have done it without the apprenticeship program. Another huge part of it that I'll talk about is we are all paid by our farms as employees, but the apprenticeship, once you get into the program, you also get a small moving stipend and you get a graduation stipend. And the farms are also given a small stipend to support your employment as well. So that's huge, right? Next slide, please. So the Burtis family has been operating off of a series of limited purpose aquaculture leases. If you got one of these Aquaculture Facts 101, it tells you a little bit about the leasing process in Maine. But we were recently granted a 2.5 acre standard lease, which is huge, and that took about 3 years start to finish, and a lot of paperwork through Department of Resources and Army Corps, as well as lobbying for social license in the area. So in Maine, riparian landowners own up to the mean low tide line, meaning they own a portion of the intertidal land in front of their house, and so they have major stake in the waters in front of their property, right? And although we do lease the water that we farm in from the state, community property owners have the opportunity to petition against an aquaculture farm for various reasons of their choosing. So it's really important that oyster farmers have good social standing in the community and get buy-in from the neighbors into the farm and the work it provides. So in my opinion, this is something that Maine as a whole is doing really well. It's generating public buy-in and creating a sociocultural shift around aquaculture in the state. And to draw kind of a perfect parallel, being an oyster farmer in Maine, I feel pretty cool most of the time. I feel kind of like a rock star when people ask me what I do for a living. The conversation really goes like, all right, what do you do for work? And I'm telling them I'm farming oysters, and that's so cool, you're providing a source of sustainable seafood. Tell me more. Whereas in California, as I'm probably preaching to the choir here, on my Uber driver from the airport, the driver asked me, first of all, what I'm doing here, and second of all, what I'm doing for work. And it not only laughed that I was going to the California Seaweed Festival, but also told me that it was the strangest Uber conversation he had ever had, hearing about working on an oyster farm. So although California and Maine are obviously very different places and have very different cultures around the waterfront, I think there's something to be gained here. And I think my biggest takeaway from working in Maine is that if aquaculture is going to be a viable climate solution and a real source of regenerative seafood, then the science isn't enough, and we have to start changing what the average person thinks about aquaculture, and even adding that word to the vocabulary in some instances. We got to keep doing things like the California Seaweed Festival. This is awesome. Uplifting the authors and artists and foodies that bring amazing light to the opportunity to farm the ocean. And we have to make it easier for fishermen and people who want to work on the water to get into aquaculture and to create pathways for young people like me to get in the industry themselves. So with my extra time here, I'll go to the next slide. Next slide, please. I'm just going to shout out some other really badass women in the industry out in Maine. So if y'all are on social media, give these ladies a follow. Corinne Newfie with Salty Talks, having conversations with researchers and farmers and foodies out in Maine talking about aquaculture. We have Amy and Alicia Galliero with Naughty Sisters Sea Farm. They have a great social media account where they're talking about their day-to-day as oyster farmers and inspiring young women to get out there and start their own farms if that's what they want to do. Libby Davis with Lady Shuckers. She's championing marginalized communities in the industry, lifting up BIPOC and queer people and making their voices heard. And finally, Ivy Barrows with Deer Isle Oyster Company, experimenting with totally plastic-free oyster gear and pushing the industry in that direction. Yeah, next slide. So that's all I have for you guys. If you want to give me a follow, I'm trying to post a little bit more about my journey as well and follow in those ladies' footsteps. I might have time for questions, I think. No, great, awesome, find me later.
Sorry about that. Thank you.
No, that's okay.
Okay, thank you very much, and thanks for understanding. We have to move on. We're running quite late, actually. The next talk is Light Matters, Decoding the Influence of Light on Seaweed Development. The speaker, Jose Avila Peltroche, Carnegie Science, Biosphere, Sciences and Engineering, Stanford.
Okay, I have a timer here, so I can just finish on time. Okay, well, thank you. For me, it's a really big honor to be here. Today, I'm going to talk about some of the research we are doing at Carnegie Science in Stanford. As I mentioned, I'm Jose Avila. I'm postdoctoral researcher at Carnegie, and today I'm going to talk about light. Light matters, decoding the influence of light on seaweed development. Next, please. So, if we go back to high school days, we learned that light is very important for photosynthesis, right? Converting light energy into chemical energy. Next, please. Light is composed of several colors that goes from violet up to red. All those different colors have different nanometers. And all these ones can be sensed by photosynthetic organisms. I'm talking about not only land plants, but also algae, seaweeds. Next. So what are the other roles of light? This is an example only with plants. First of all, adaptive responses. For example, when plants sense high light intensity, they might change their physiology to adapt to these new conditions. Developmental responses, different light colors might trigger, for example, germination, floral transitions, and death. And also cellular and subcellular responses, for example, chloroplast movement. Next. Here, I'm going to focus on developmental responses, and because we are in the Seaweed Festival, I'm going to give you a really nice example of how light quality, which means colors, can affect these kind of responses on seaweeds. Next. So this is Undaria pignatifida, wakame. I worked with this seaweed for 5 years on my PhD back in South Korea, and I know some of you might know this kind of response. If you keep for example, the female gametophyte in red, this gametophyte is the microscopic stage, will just grow, grow, grow. You can even grow it in bioreactors. You can even extract some compound from the gametophyte itself. But once you switch to blue light, then you will induce reproduction. They will produce these structures, you can call it eggs. And after fertilization, you will have a young sporophyte that later will go into the ocean, will grow as the delicious wakame for the seaweed soup. However, my question is, okay, this is well known, but what is happening behind? What are the pathways? What are the genes that are activated under these different lights and other kind of lights? Next. Then my question is, how light is perceived in general? And when light modifies some kind of behavior, this is directly absorbed by protein molecules called photoreceptors. This is a very nice 3D structure of a photoreceptor, but I simplified in this diagram here. Basically, a photoreceptor is composed by a protein part and a chromophore, which is a small molecule that can sense different light qualities. Blue, green, red, far red, and even UV. Next. Another question. How light change underwater and how seaweeds can adapt or respond to these light variations. This is a really nice video from the kelp forest in southern Peru. And next. And okay, you might say that, okay, the ocean is blue, right? Or not? Is the ocean blue? The open ocean, yeah, looks blue, but if we go to coastal waters, it's not that blue. It's kind of greenish sometimes. It depends on, the environmental conditions. Next. And here we are talking about specifically light penetration. If we take the open ocean and coastal waters and compare the colors that can penetrate, we can see that blue is the one that penetrates deepest compared to the other colors. However, in coastal waters, in the green region is the spectrum that penetrates deepest compared, for example, to red or blue. And this is because in coastal waters you have phytoplankton, you have detritus, you have many different things that can absorb these other light colors. Important thing, 1% of original light at the top is left at 100 meters. So actually water is really good at absorbing light. Next. Just a reminder of when we are talking about seaweeds, we are not talking about only one single group. We are talking about red, green, that are mostly related to land plants, and brown, that is another story. They are related to dinoflagellates. They are related even to, I usually say that, Plasmodium falciparum, which is a pathogen. So they are really diverse. Here, I'm going to talk about algal photoreceptors. Next. There has been many studies, actually, in unicellular algae about this topic. I can mention here some examples. For example, phytochromes that can sense red, orochromes that are specifically, for example, for brown algae and diatoms that sense blue, as well as cryptochromes. Next. And now I want to talk more about brown seaweeds, my favorite group of algae. Next. What do we know about light quality on brown seaweeds? Actually, there are quite good studies back in the 80s, for example, testing different light colors in developmental responses, physiology, photosynthesis. I already mentioned the most common example, kelp reproduction, red, blue. However, again, do we really understand what is happening behind? What can we do to understand what are the pathways that all brown algae share in common? One option is maybe we can study all the brown algae species and start to understand the different pathways. That's a really nice possibility, but actually it will take a lot of time. Another option is to use something called a model organism. And today I want to introduce you to, next, one more. Exocarpus, my new favorite species. Exocarpus was proposed as a model organism back in 2004 for developmental biology, and it's a model organism for brown algae. This is a small petri dish from the lab. It's probably like 5 centimeters long, and these tiny hairs are Exocarpus. It has a simple morphology. It's easy to handle in the lab. My question, can you handle in the lab the whole life cycle of, for example, bull kelp? Complete the whole life cycle? It's quite difficult. It's possible, but it's quite difficult. And it will take a lot of time. You can close the Ectocarpus life cycle in 2-3 months. It's really good for genetics and cytology. We have the genome, the transcriptome, and we have even genetic modification protocols available that we are also testing. Next. So this is our beautiful Ectocarpus. It might look simple, but it has some really interesting characteristics. Next. We have basal filaments that use Ectocarpus to attach to the surface. And from those basal filaments, we have, next, the upright filaments. And the upright filaments will form, next please, the sporangium, which ultimately will release, one more please, spores. There's a small video there from spores. This spore will settle down and then will grow as a new ectocarpus. Next, please. So according to the ectocarpus genome, they have phytochromes, autochromes, cryptochromes, all those photoreceptors that I mentioned. Next. However, little is known about light quality on ectocarpus. I mean, if we expose ectocarpus to different light colors, what will happen? We don't know. And this is the main topic of our research at Carnegie. Here, next, I'm gonna show you just one preliminary result. And I invite you all to visit our poster. We have more data there. This is one of the first experiments we performed, like cultivating ectocarpus at different lights. And what can you see here? Let me see your time. Okay, I'm good. This is the normal morphology of ectocarpus. You know, basal filament, upright filament, and then of course the sporangium. However, if you go to the red, almost going to the far red, you are kind of first inhibiting growth, but also, for example, in the green, you are inhibiting the formation of these upright filaments. Even this base is growing a lot, it's growing and growing and growing. Somehow, there's something missing there. What is something? We think that the blue light is really controlling this transition from the basal system to the upright filament and, of course, later on, reproduction. Next, please. I want to finish this presentation explaining why are we studying light quality. This is a basic research. So we have talked about aquaculture, restoration, applied things, and mostly studying basic research. So why? First of all, obviously, to broaden our understanding of photosensing mechanism, which is key for the second point, controlling the life cycle of the kelps and in general bronalgae to improve our current breeding techniques. This is very important, and I personally think that it's something that is lacking nowadays. Do we really understand what we are culturing deeper? And finally, and this is one thing that I was thinking during the last weeks, we have some problems also with bronalgae. Well, you know the sargassum. But some people that, for example, cultivate red algae, they might find some of these brown epiphytes. It's very, it's a very interesting problem, and we hope that with our results later on, we can prevent or mitigate these brown algal blooms or epiphytes, because again, light is controlling growth and development. Next. Thank you, everyone. This is the team. Well, that's me. Frey is over there. Andrew and our PI, Arthur Grossman. And thank you to Carnegie, especially to the Grossman Biolab. Well, if you have any questions, maybe we can talk later. Thanks.
I think we've got time for one question. Last talk, and thank you all for hanging in there, and thank you for your patience. The last speaker is Ashley Kidd, who will tell you a bit about a community-based project to restore sunflower sea stars at the Sunflower Sea Star Lab.
All right, thank you for hanging out to the last session of the day. It's been really awesome to listen to all the talk about kelp, so I hope you're ready for a kelp-adjacent talk with sunflower sea stars. We are growing these little sunflower sea stars. These are some juveniles that I will get into in our talk. So next slide, please. I work at the Sunflower Star Lab, so just to give you a brief background on who we are and what we're doing. We started as a nonprofit in 2021 as a result of a request to expand sunflower sea star culture work outside of the wonderful but very small lab up at Friday Harbor Labs at the University of Washington. We are currently located at a public-facing lab in Moss Landing, and we have wonderful interns and volunteers, as well as our lab manager that run the lab 7 days a week. And we couldn't really do it without the community of Monterey and Santa Cruz and Moss Landing. So we have public outreach events, and we try to involve the community as much as we can. You can literally walk into the lab if you're going on an eco tour or having lunch at the restaurant across the way. So it's a really fun way to engage our public and expose them to the culture of a critically endangered sea star. The little guy in the middle is one of our sunflower sea stars munching on a purple urchin. Yay. Yeah, so we do everything from growing the microalgae. We've got larval culture systems for urchins and sea stars and grow out tanks. And yeah, a lovely little group of volunteers. Next slide, please. So what is a sunflower sea star? How many people have seen a sunflower sea star? How many have seen them in the wild? Ayo, wow, this is the right group to be to be in. For those of you who haven't, it's sad that they really haven't been around in California for the last decade because of a sea star wasting event. But if you came across one, and you can see them in Washington State, and this is what a sea star looks like. A sunflower sea star looks like. They've got multiple arms. They can get over a meter wide. They're related to urchins and sea cucumbers, crinoids, and brittle stars. And they are one of the sea stars that exist off of our Pacific coast. Next slide, please. And they exist everywhere. They're not just kelp forest inhabitants. They are found in seagrass beds, under docks, in kelp forests, and sandy or mud bottoms. But they're also, next slide, generalist predators that used to exist from Baja in the south all the way up to the Aleutian Islands in Alaska. But in 2014, or late 2013, early 2014, a sea star wasting epidemic started and spread very rapidly over a course of a few years from California all the way up to Alaska, and by 2018, they had lost 95% of their population. So we are in the orange in the north coast of California, and if you look at the numbers, that's the percent loss of sea stars. So all the ocean coastline south of Washington, or the San Juan, or Strait of San Juan de Fuca, or Strait of, yes, thank you, have become functionally extinct along that coastline. So there are no numbers to fulfill that ecological role anywhere, kelp forest included. What does that mean? Next slide, please. This is unfortunately not an uncommon sight here in Northern California. A lot of the kelp forests have undergone a lot of stressors, one of them being a loss of a predator that can eat the urchins, and urchins graze on kelp, and we're all here for the Seaweed Festival, and we love kelp. And we hopefully, after this day, have learned of all the benefits and needs to bring kelp back to our coast. So this is the sad state of a previous kelp bed that is now an urchin barren. And these urchins don't really have that much food to munch on. As you can see, they've kind of munched it all away, and they're just kind of hanging out. Next slide, please. So sunflower sea stars are one of the major predators. Yes, other sea stars do in fact eat urchins, but these are the largest, fastest cheetahs of the intertidal or subtidal for as far as marine invertebrates go. What we've noticed, or what other researchers have noticed, is that not only do they have this predatory effect, but of eating them, but also the behavioral effect of just having the presence of a predator in an ecosystem changes the behavior of urchins, and that could be a net benefit for kelp recovery. Next slide, please. And so when I talk about these other people, this is one picture depicting not 100%, but a good number of people that are part of the Pycnopodia Recovery Working Group. This began in 2018 after this epidemics, tore through our continent, and they brought together a couple researchers and aquariums to ask, what do we do about this problem? And so that's when the Pycnopodia Recovery Working Group began, and the Nature Conservancy sort of took the lead on convening researchers every few months to start answering all the questions from behavioral ecology, disease ecology, genetics, what's going to happen, aquaculture. And so you might see a couple people in the audience in this picture. It's a wonderful group of people that we get to work with. So it takes a village. Next slide, please. So from that, this group developed the Pycnopodia recovery, or the roadmap to recovery for the sunflower sea star. This is our roadmap on all the different steps that we need to address to recover sunflower sea stars. It's got 7 objectives, everything from outreach to genetics, how to culture them. And from that, the Association of Zoos and Aquariums, we were able to propose the species as a species under the Saving Animals from Extinction program because it has a recovery plan that was published in 2022. So in 2023, we became an official safe species, which gives us opportunities to access funding from the Association of Zoos and Aquariums, which again, for a species that has to compete with all these other wonderful, important recovery issues, we now have a funding line that we can access because we've been acknowledged by this association. And then most recently, as of this month, we received a 3-year funding from the NOAA Transformational Habitat and Restoration Coastal Resilience Grant, so that we can be part of the full ecosystem recovery planning. So we're trying to remove the silos of species recovery as individuals and bring kelp and sunflower sea star and abalone recovery together. And again, there's so many people that have been cross-referenced in all of these talks today. It just makes sense that we're all working together. So it's been really wonderful to see that come to fruition and be acknowledged formally. Next slide. Or just, dance interlude. So how do we do that in California when they are functionally extinct in the wild? Well, this comes back to having public aquariums that were able to keep alive a very, very small number of sunflower sea stars in public aquaria. These were a couple institutions, the Vancouver Aquarium, as well as Friday Harbor Labs, Oregon Coast Aquarium, Henry Dorley Zoo in Omaha, the Aquarium of the Pacific, and the Birch Aquarium at Scripps were the aquariums on the continental U.S. There are also sunflower sea stars at the Alaska Sea Life Center and at Aquarium du Québec on the East Coast in Canada. But we were able to start immediately looking at what the spawning events were like in all these institutions, and then start to note whether they're male or female, because they don't have any sexual dimorphism, so we don't know which ones are male or female. And up until this point, nobody really had a need to know what they had in their population. But because of that, this is actually an image of one of the males spawning at the Vancouver Aquarium. So the nice thing about aquarium is you don't have to dive in a wonderful but sometimes harsh ocean environment. You can just walk in one morning and watch a sea star spawn. And that's very convenient. So by 2023, we'd got most of the broodstock sexed. And then, next slide, we actually, we're able to identify a male and female at the Birch Aquarium. So this is the Birch Aquarium at Scripps, where the Cal Academy of Sciences, Birch Aquarium, Aquarium of the Pacific, Sunflower Star Lab were present to spawn them. And this was the first time we had successful fertilization in California on Valentine's Day, 2024. There was a lot of dancing and screaming happening because one of the issues is because of the nature of the sunflower sea star wasting disease, or transfer across state borders is not feasible at this time. So we really had to use what we had in the state to just attempt to try to use these animals as research subjects in California. There was really no other way to do it. So we had a great time at Birch, and from that we were able to distribute larvae to 5 different institutions, all up and down California. Next slide, please. And as a result of that, there were also other successful spawns at the Vancouver Aquarium, at the Omaha Zoo, and this was the first gathering of participants who'd actively assisted in raising sunflower sea stars in the world, or sea stars of any kind. So that was a really exciting thing that we got to do in May, to come together after a few months of having raised the larvae and settled the juveniles, and sort of have everyone come together and take next steps and sort of share what they've learned, and also go back up to Friday Harbor Labs and figure out, go back to the source where it all started and be inspired to move on and answer our own questions. Next slide. So this is the life cycle of a sunflower sea star to date. This image was the last image on the bottom as a juvenile, 8 months old sunflower sea star taken at the Sunflower Star Laboratory at Moss Landing, devouring a nice little purple urchin. But they do have a larval stage that can last a month or many months, depending on the environmental conditions. And then they settle out into a 0.5 millimeter little baby sea star, and then very, very, very quickly are ravenously eating whatever is appropriate to feed them, and they keep growing and growing and adding arms and eating more and more every day, it seems. Next slide. So there's a lot of messy data measuring a sea star that's very amorphous. It can stretch and recoil. We have like some interesting size measurements. You can measure a sunflower sea star like 5 arms and then measure another 5 in like a minute, and they'll be a different size. So they've been really fun to watch. They're super active, but they are definitely growing no matter which way you slice it. And it's been really fun to watch those grow. So right now at this very moment, we have 8 institutions, Cal Poly Humboldt being the most recent recipients of our sunflower sea star juveniles. Woo! And we have 314-ish juvenile sunflower sea stars at different institutions across the state that researchers are now able to answer all the questions that they're best suited to answer. It's been really great. We've gone from a state that had no sunflower sea stars in juveniles to 8 institutions and hundreds of them. So it's been a wild ride so far. Next slide, please. So I had bringing up babies as a title at the beginning of my presentation, and this is a wacky movie from 1939 with Katharine Hepburn and Cary Grant, where she was raising a leopard in her beautiful, wealthy suite. And not that we're treating them as pets or have named every single one of our sea stars, but they are kind of like raising they are cheetahs of the sea. And it is like, next click, they're like raising a leopard. They are ravenous. They are predators. It's very cool to be working with. I've worked with marine invertebrates before, but abalone eat algae. So it's really interesting, and it's a really interesting restoration question to understand where the trade-offs are on raising these animals and how to go about the recovery. Because you do have to culture urchins or food in order to feed these ravenous hordes that we have. Next slide. And this is just the beginning. There's so many questions that we want to answer. Next slide. So if you guys are out in Trinidad tide pooling, you can definitely do your part and take a picture of a sea star if you see one and send it to us and put it on iNaturalist. Join Reef Check if you're a scuba diver and want to do citizen science, or post on Marine and track the disease events, because that's really important to understanding how we're going to go about recovering the species. And just go out there because they've been seen in Trinidad. Next slide. This is a recent post of the sea stars that have been pictured and tracked. That's Trinidad, where you guys are sitting right now, and a couple more in Mendocino. So that's not a recovery by any stretch of the imagination, but they're back in California, woo, as little baby sea stars. So looking at how they're going to react in the wild and where they're responding, I thought it was really cool listening to Marina's talk. There you are, you moved. Earlier, just looking at the salinity and like why certain areas have good bull kelp growth and no urchins. You know, it's like, why are the sunflower sea stars deciding to settle in these areas? You know, so lots of questions that we can now start answering in the field. And that's it. So thank you to all of the partners, so many people who've supported us or talked to us, and working with Cal Fish and Wildlife. Yes, so much. So many conversations, building trust, and just, you know, reaching out to the community to work with everyone. It's been really wonderful, and thanks for coming.
Thanks for being here. I'm sure the speakers who are still in the room would be happy to answer your questions, but we'll let you come up to them right up here in the front. Thanks again to all the great talks and speakers.